coming from macroplastics who have been weathered or degraded or partially broken due to the effect of light, the oxygen, mechanical abrasion, even also a biological broken. For example, a paper published by a Chilean colleague when he was doing her postdoc in Australia report that crabs are able to break these plastics, digest the plastic, and in the fecal pellets they appear like fibers. So as part of the biological or the daily life. But the thing is that macroplastics have one effect. Most of them is a level of, for the bigger animals, like entanglement, like gut blockage, for example, what we see in whales. But microplastics is another different history because first of all, they are not easily seen. You need to employ a logistics to study them and just pray to find them in your samples. You know, it's not easy to find them. There is, once microplastic reach the ocean, we have different processes. Microplastics are able to be part of this mechanical degradation for the waves, as mentioned before. We have biofouling. This plastics represents a plastisphere, and microbials start to grow. So it changes the density, and they don't float, so they could be in the mid-water column or in the deeper water column. And also, they could be eating and could be part of the fecal pellets of copepods, for example, affecting also the carbon cycle in the water column. And then the other part that's very interesting to me is the weathering from ultraviolet radiation, which is quite important, especially on plastics. You go to the beach and take one plastic who was exposed during a lot of time, and you try to break it, it's more breakable than the new plastic. And this is the part that really interests me as a scientist. It's how they are able to enter the food web. How are they transported through the food web? And how are the things that tell us information about this accumulation? Because, as Cara said previously, there are a lot of reports of ingestion, but ingestion doesn't mean trophic transfer, I think. And one example is that we eat a fish, but we don't eat the stomach of the fish, which is presumably where most of the plastic will be accumulated. But in the case of marine communities, they eat mostly the entire animals, except in the sharks, because they bite. But usually they eat the entire fish. The prey is the entire fish. And this is like a study just telling us, this was Ungo et al. In 2021, just reporting what we do study in the water column. What is the animal group that we study usually? And most of them usually are the fish. And in fish, we are focused usually in the gastrointestinal tract and the stomach. Because we have a bias there. We think that every plastic should be there. And we have some experiments that I will not show here, just reporting how they accumulate inside the animal and the effects on the digestive tract. And the others are the invertebrates. You see the water column, the two most important steps in the water column in the food web transfer are fish and invertebrates. Also phytoplankton, but plastics and phytoplankton would be a problem because they aggregate and they bond, but there are not too much reports on the phytoplankton aggregates in the open ocean, so it's very difficult. So we could assume that they are floating around, but there is no proof that this could occur in the natural ocean. In experiments, there were some experiments performed in the university in Germany, in Kiel, that they show that the aggregates are able to modify the phytoplankton performance and to affect the primary production, which is the base of the trophic web. So we have different trophic levels, and all of them are connected. There is no a linear connection because, for example, this big fish could consume here, 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 even here. It depends of the feeding strategy. But the problem is that we don't know the role of plastics here, and we know that this kind of animals could have plastics inside, and also this. So the food, or to feed on the lower trophic level, entails consumption of energy, entails uptake of carbon, and also classic pollutants, for example, as I mentioned, persistent organic pollutants like polychlorobiphenyls, polybromodiphenyl ethers, and pesticides. But also those emerging pollutants now in environmental chemistry, plastics are believed to be emerging pollutants. Because what is an emerging pollutant? It's something that we don't care 20 years ago, that exists in the media. So we published a paper, all the researchers working in Chile on plastics, just looking to what we have done in the last years. And it was a very good experience because we looked to each other. What are you doing? What I'm doing? What can we do together? And similarly to the graphs that you see on plastic production, you see an increase in the number of articles. I think the numbers are not, but we are a small community. So when we look to what kind of plastics did we study, we were centered in the microplastics, especially in the last 5 years, because we learned during these years how to study microplastics. We learned, we got the technology, because, for example, Cesar told at the start that most of the studies are centered in the Northern Hemisphere. But our problem was the access to technology. I remember that 6 years ago, all the researchers of this network have access to one FTR. Because material scientists have FTR, but they were like, do you want to touch my FTR? Really? So we built our own libraries because libraries were expensive, and say, okay, we don't want to buy a library, we will construct our library. And now, for example, in my research group, we have one micro FTR, which is working well. It has 2 months in the laboratory, but we are doing every day working on that because we are starting to do a lot of things, especially with grad students who come to the laboratory. Just they want to work with microplastics. So here is the equipment. Let's do it. And the other thing is that we start to collaborate with people from the Northern Hemisphere, because we knew each other in the first Congress. For example, if you look to the papers that we published here, we say, okay, plastics are blue, and they are round, and that's all. Okay, now we see plastics are made from PVC, or we have polypropylene, or we have PIT, and also we are able to study, and they are full of polybrominated compounds that are flame retardants. So there was a change. Also, I don't show usually this graph, but also the impact factor. We hate the impact factor, but it means something sometimes. The impact factor of the journals that we published increased because we increased and we got better doing our job. When we look to what we reported in Chile, these were the main topics, which is the entanglement, the ingestion, epibionts, dispersal, contaminant and microbes on microplastics, sea surface abundance and distribution, shore abundance, and review and social survey. This is one of the most important in Chile. I don't know if you know Martin Thiel. Martin Thiel is one of the, I think, probably will know the number of científicos de la basura. This was an initiative performed by Martin Thiel, and just working with the schools and just to report the plastic, where it comes, not only the plastic, but also the debris that appears, where it comes, where it appears, what is the occurrence, can we do something? Not just taking out from the beach, trying to make people to understand how it behaves in the environment, this debris. But ingestion, as you can see, because the entanglement was this part here. But as you can see, as I told you, we increased our knowledge and we start to report the ingestion. Because we knew more about that, and we knew better how to do that. Ingestion was reported in the whole coast of Chile, from different worlds. Most of them were on fish, of course, but also marine mammals. And also, we found this in Antarctic fish. It's in preparation. Sorry, I cannot show anything yet because we are trying to understand our data. But we found, we take about 100 fish, like this size. They are called mactophids. They are very common in the whole ocean. And we find plastics in 10 of them. But when we took samples from the coastal Antarctica, near the human settlements in Antarctica, the numbers increased a lot. I think we found plastic almost in every fish that we. But you must think that it was the Antarctic island who has most presence of humans. We have King George Island is like the entryway of Antarctica for all the research stations and also for the tourism. So it makes sense where there is a lot of people hanging around, you will find a lot of plastic. So now I want to talk about where we found plastics, okay? Just to realize how this transfer could occur in a food web that we know very well, which is a food web from Waffle Island. Waffle Island is an island with a big colony of otarids. I think it's the biggest colony of otarids in the South Pacific Ocean, and it's the northern one. It's like here. And in the island there are only otarids and army people. There's something, 5 military, and then like 1,000 of otarids who live there, who raise their poops there. So we started to look for what they eat. And what they eat is king crab. Most of you will know because it's a very valuable economic source, especially for local communities. And what the group of Andrada Novando from the University of Magallanes found is that they found especially fibers. Most of them were passed through an FTR, and we say that most of them were made of nylon, and the materials that belongs to the long lines that the fishermen from the area use it. And that's what quite interesting. So we know that king crab from the deeper sediments, it's plastic, or at least contains plastic. What about the smaller guys? We don't have any report from Chile identifying copepods or similar to copepods, this kind of zooplankton, eating plastics. But we know that from other areas that usually appears. So this, I think all of us know these images because we saw them probably on social networks. And not only they are able to eat them, but also they go with the fecal pellets and go to deeper waters, affecting the carbon cycling. But we know that these animals are able to eat and to retain them inside. Now we are going with fish, because fish eat copepods, or another fish. This was one experiment from the group of Martin Thiel and Nicolas Sori, a postdoc which is now in France, when they saw that fish are able to select their food because of their color. So You have copepods in the environment, which are blue color, which are the smaller ones, okay? You see microplastics blue. You see that fish feeds preferentially on the blue. This is what they found. And we have like an inverse correlation. What you found in the fish, what you found in the copepods. That's quite curious. And this is what they look like. This is so curious, no? Because one person would think, but did the fish see blue like us? This is one of the things that that was discussed, but it appears that from the physiology of oceanic fishes in general, we don't know about the specifics about that, oceanic fishes are able to distinguish from black, white, and some transition from blue to green, and they are not able to detect red or yellow, on the contrary to coastal fish. When they were able also to differentiate, this is the first work that we performed on microplastics in my group, and they were able to differentiate red from yellow, from blue from green, because of their feeding strategy. What we found here is that usually carnivores feed more on red and orange, herbivores feed more on brown, green, and something blue, and omnivores feed on everything that appears there. Because they are omnivores, they only need to grow and to eat and to eat and to eat to grow, because there is a part of their life in these tidal pools, which is areas that are very extreme for them because of the wave movements, and they got full of water, and then 2 days later they were run out of water, so it's quite difficult. And that was so important for us to detect this. And the problem with the fish is that most of the ingestion studies report the occurrence of microplastics in the digestive tract. We don't know if it is because of feeding or because of respiration, because they use the same mouth to take oxygen from water. We also exclude them, but this is not usually. I think when they feed, the digestive tract is blocked. You know, because we feed, we block the digestive tract, and then we do respiration. So it's a different way. But sometimes there are some reports of plastic fragments in the gills, similarly to what you say about mussels, because this is a filtration system. And when it appears, they might be involuntary. But we agree among the researchers that usually the consumption of plastic is because of confusion, because we don't know if it's plastic or no, the fish, but we need to eat, so in case of that, just buy them. This is what the animals do in these kind of things. Let's go to marine mammals. These were otarids from the study area, from Waffle Island, and we found these clearly macroplastics. Most of them were also composed by polypropylene and some of them PVC and nylon, but in the area there is full of salmon culture. And nets is what they use more. And if you look to the colors, they are blue, because the nets are usually blue or green. It could be, the color could be changed because of weathering. We tested feces, because you are not able to take a knife and to open a, no. I think the ethics committee will kill us. So we use this indirect thing just to look at the feces, and of course, if there are plastics in the fecal pellets, it probably will be plastics inside the animal. Okay? And there was one experiment conducted during this year also in England with in a zoo, when they gave plastic contaminated fish to animals in captivity, just to know what enters the animal, what gets inside, and what goes outside. This was a very interesting work, because with these numbers we could modelize how plastic behaves. You will understand later why. also we found this, not latitudinal gradient, I didn't put the latitudes, but this is Peru, this is Juan Fernandez, which is proximate to the South Pacific Gyre. This is Pupuya Islet, which is very near from Santiago. Guafo Island, Chulec, and Punta Chayhuaco, which are in the Patagonia, and we see that islands usually, especially those islands that are inside the ocean, they are not very proximate to the coast, have a lot of plastics. Because of oceanic transport will accumulate them. And also they have a deficient waste management, because it's an island. And Guafo Island was our area, but also we find levels of microfibers which were comparable to what we found in the island. So this is our animal. This is our case of study. And one of the things that for us was very important is not only to report if they are feeding plastics or plastics have some occurrence inside the animal, is where did they come and where did they go? So this is not only to know the concentration inside the animal or the levels inside the animal, it's important to know how much they feed and how much they digest. So this is one of our main questions that we are working now. And for this, we use a similar approach that we use to persistent organic pollutants. Everything is about the ratios, induce ratio, uptake, digestion. We could argue that could be some biotransformation, but not likely to be. Biotransformation is usually for more degradable compounds like plastics, like aromatic hydrocarbons, for example, that are more easy to degrade than a plastic. And there will be a growth dilution, but this is not the case. I will explain later, because plastic, in the best of the cases, have a half-life of days, from hours to days. In a copepod, probably the plastic will be some hours, is the transit time. And in these animals, the digestion takes 1 day, more or less. In whales, takes like weeks. And this is important because when you want to study the uptake and elimination processes in an animal, you have the loss for reproduction, but this is likely not to be, because plastics could be transferred, but it's not very, there is not much proof about that. But if you want to know the concentration, you know what they ate, and you know what they eliminate, then you're able to reproduce this through a food web. So you will know, okay, everything is fish, but we'll do a sketch. This is a marine mammal, this is a small fish or a crab, okay? So if you want to know what is here, you need to know this one. But you have some limitations, that is the uptake. You don't know the uptake. One of the things that is important is, okay, I have this Mike Toffee, who eats some copepods, for example. How much copepods eats per day? How much biomass it's per day? And inside this biomass, how much was the percentage of microplastics? So this is important to take into account. And this is what we are doing now. And the problem with this is the logistics. You must think that we travel 4 days to Guafo Island, or 5 days, my students, not me. And they spend like 1 or 2 months there working, but these sea lions doesn't stay there. They move around, especially the females during the growth season, during the rising season. And the other thing is that you need to collaborate with the fishers from the area. Just give me some fish that you take. You buy usually them. They don't give you give you for free. But now we are trying to figure out how to estimate this, because we saw one paper working with salmon, and they know, because salmon is a very important economic species, so they know exactly, not exactly, but with a good confidence what they ate, how many they eat, everything. They know everything about the salmon. So they were able to predict how many plastics will be inside the salmon, and how many plastic will be outside, and how many will be the uptake. So we are trying to replicate this with our limitations, and now we're planning the next year field survey just to try to estimate, because you never will get the final number. You will get the estimates of that, and you will have a ratio of confidence just to know and to figure out how plastics are transferred. But as far as I know, there is no a characterized food web with this plastic transfer. And I know because of that, because there is no information, is because the logistics are very complicated, and also your number of samples should be very high just to take confidence of what you're saying is not like a photograph. Just you need an album to prove something, not just a photo. So I think this is our main message. We want to working on that, on bioaccumulation. We are doing it now, and we are doing it in the end of the world, in the Patagonian region. So that's all, and thanks you for attending in the morning.
There's time for 2 questions if there's any questions in the audience. Otherwise, I'll start with one. There's one? Okay. Felipe, here you
go. Hi there, I'm Felipe, a student from Caesar Lab. And I would like to know about the state of the art in the impact of the plastics in mammals or birds in the marine environment, because I read, I don't know the year of the paper, but I read that they were estimating, I don't know how to say this, estimando?
Yes.
Well, yeah, estimating like 1 million mammals and birds from the marine environment dying annually because of plastics. I don't know if that number is correct or if there is another estimation now. So yeah.
No, but
thank you.
I think I probably heard about what you said, because there was one study, for example, looking to birds from people from Australia, and looking what are the deaths based on the occurrence of dead animals retrieving. But you must think that these studies are based on data reporting, and this is the same that Cara said previously about the production of waste for the countries. It depends on the data, but I think it's not important to take about numbers, because you say, oh, there is 1 million birds dying, or there is 100,000 animals dying. I think more than the exact numbers, we need to look at what are the process that is affecting that animals, and what we can do to avoid this. For example, when people talk to me, okay, but plastic will be easier. You just take them. Like the approach of Bojan Slat, for example. They take him, and he will solutionate the problem. The problem is not to collect them in the open sea. The problem should be here in the land, no? And going to your question, for example, we are working in a report. Now I expect to submit the paper by the next month, working on the impact of on otarids, not only otarids, also in falsids, o sea, focas y leones marinos, around the world. And we work with the Van Seville model of the larva, because plastics could behave similarly to a larva, and also with the reports of ingestion and entanglement, and also with the fishing effort doing by each country. You know, there is reported that how many boats are going to fish somewhere. So you are able to estimate the impact of this one. And despite of the number, for example, in Hawaii, there are a lot of animals stranded with plastics around the neck. In Chile, we have 10 in 60 years, for example, in Guafo Island. So it's not about the numbers. It's more about to understand the process that are affecting that animals. Once you understand the process, you will be able to tackle the problem more correctly. I think.
A while ago, I was reading some reports that, because you were talking about also persistent organic pollutants, some pollutants which are present in the marine environment can accumulate in the fatty tissue, especially of animals of higher trophic levels, such as whales, porpoises, whatever. And we have previously heard that some of these compounds can sorb onto plastics. Is there any evidence that such compounds which sorb onto plastics can actually be transferred
there is a lot of
animals?
there is a lot of evidence. I will show you. There is a global network of passive samplers of pollutants in water, and we use polyvinyl acetate to collect the pollutants because they equilibrate. I think now we are going to change to chemistry because we are talking about the partition coefficients between water and plastic. So you have one pollutant, let's say naphthalene, which is very volatile and is not very hydrophobic. It will be more soluble in water. But let's talk about pyrene or benzoapyrene, which is more hydrophobic and is not very soluble in water. So when we want to measure these compounds in the water without electricity and with a very simple logistics, because when I sample air in Antarctica or water in Antarctica, I think we talk about a lot about our logistics, but we go with very big equipments. We need a lot of electricity, but the passive samples are made of plastic, most of them, because most of these compounds partition preferentially to this plastic to water. So we use them to measure and to give and to have an idea of how the amount of pollutants that are floating around. Also, there are some research groups that are doing this on animals. They put a plastic strip inside the skin of the animal, and they were able to predict the concentration of the animal, just measuring the strip, which is less invasive to kill the animal and to take a sample. And this is based on fugacity approach, which is classical in physical chemistry and environmental chemistry. But yes, you have a lot. For example, one other example, we sampled plastics in the Corcovado Gulf, which is in the south of Chile, and we need to dilute the samples for PBDs up to 10 times because it was saturation, our GCMS, because probably they came from an installation who needs a lot of flame of retardant, or the plastic has a lot of flame of retardants. But we need to dilute because the equipment was, when you're seeing the signal, they go far, 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 and the equipment started to not working very well, so we dilute. For plastics, we dilute a lot of samples. And it's in a semi-pristine area, which is the Patagonia.
I'll do just a very quick last question, and it will be maybe an opportunity for self-criticism as a scientific community. And the question will be, we are always described that we are a very long, narrow country, right? Which means in the end that we have a lot of coast. Yeah. So in your opinion, do you think that we're doing enough in terms of a scientific community to try to sample these very diverse areas all across the country to try to understand better what's happening with plastics and additives?
The thing with the plastics community in Chile, we have a network, which is called the SPLASH, Scientific Plastic Alliance from Chile. We will have a meeting on Monday, online. And we are a small community of talking in PIs, in the principal investigator, we are 15. There are a lot of students starting now. But the thing is that, for example, when Cara talked about this meeting who was conducted in Uruguay and now will be in Paris this year for plastic, we don't know which is in charge in the Ministry of Environment about that. But even also, I have been working in pesticides and organic pollutants, and I don't know the people in charge of UNEP, because when I send an email to the contact that they don't answer the emails. But this is not the problem, because finally we got them. The problem is that when you are talking about policies in Chile, what are the main environmental problems that you have in Chile now? We have heavy industry pollution, you see Quintero. You have urban pollution. So, our invited people come in summer, but if you come here in winter, it's like London. I think there is a lot of air pollution in the city. We call this smog, because it's smoke plus fog, and you will know this when you breathe. And there is one of the other main problems the other problems for the Ministry of Environment and Agriculture, the water, there's no water here. The wastewater treatment. So when you are going to the policymakers and say plastics, they say, okay, but, and they have a lot of problems. They need to solve, I need to take care. But in the case of plastics, one of the things that was most important is because we have the first international law forbidding the single-use plastic. We were the first country in the world to ban the use, but it's banned in the law, but not in the practice. Just go outside the university and you will take single-use plastic easily. But the thing is that this policy came from the down, from the people. I think this is most important. In the case of plastics and the plastic pollution, most of the policymakers are pushed by non-governmental organizations, by local communities that say, okay, we should stop this, and then it starts to go up in the policy. This is the exact case that happened in Chile. And because usually when you are going to top-down policies are more like environmental impact services, other things that are more in the big area, in the big economy, you know. But this kind of laws which comes from local communities, I think the first normative that appeared in Chile local was in a small, I don't know how to say, a small town. Say, we don't want to sell plastics anymore, and they forbid locally. And then this start to go to another areas, and now it's expanded to all of Chile. Now, for example, if you go to the supermarket, you are not able to take one plastic bag. If you go to Europe or the United States, plastic bags goes around everywhere. So this is the case here.
All right, well, thank you so much. Oh, I'll use this one. Thank you so much for the talk. And now we move on to... Okay. And so we now move on to the last talk for this morning, which will be perfect as a transition for what we're going to be discussing in the afternoon. So we've been talking about plastic pollution, the presence in different environments, but then we already know that there might be some potential alternatives for biological, at least degradation, potentially recycling of plastics, some of them, not all of them. And so for a transition, we will have Beatriz Diaz, who's a professor from the Catholic University of the School of Biological Sciences. We started collaborating 4 years ago, I think, something like that. And she's an expert in what will be metagenomics and collecting samples from different marine environments, especially Antarctic polluted environments, but also like every type of extreme environment that we have in our country, which we have many. And taking samples is trying to understand what's the ecology of those microbial systems. She got a PhD in the Universidad Autónoma Barcelona in Spain. So this is the second speaker that is actually working in Chile, but it's from Spain, which is quite exciting and quite a trip. But she's been around for a while. We've been doing a lot of collaborations, and one of the things that she's going to be discussing today is something that we work as a collaboration, but it was mainly driven by her extremely well-known background on metagenomics and omics in general and microbiology. So Beatriz is going to be talking about a global metagenomic survey of polyester hydrolases, and that's why I'm saying this is going to be a transition for this talks in the afternoon, especially in marine environments. So thank you so much for coming and for organizing this symposium.